Welcome, class. Thank you all for having a seat and getting your notebooks out and being ready to learn. Does anybody have any questions before we get started today?
No.
Yes, Ms. Arta.
How are you doing today?
I'm doing well. Thank you for asking. Okay, so today we are going to be covering the microscope, and we're going to learn a little bit about what we do with the microscope, why it's important, why we use it in science, and a little bit of history about it. So our objectives for today, you should be able to effectively use a compound microscope to identify specimens, specifically cells. That's going to be what we move into next, is cells, different types of cells. You're also going to be able to identify the parts of the microscope, so to know their function and what they do. That's really important. And I also want you to know the difference between a prokaryotic cell and a eukaryotic cell, okay? That's really, really pivotal in our next content that we're going to do, hopefully at the end of this week, okay? So, to begin with, let's talk a little bit about microscope handling and microscope safety, okay? So, the microscope has what we call an arm, which is right here. These conveniently have a handle on them for you to be able to pick them up. These are pretty light, but they're very expensive, and we need to be sure we take care of them, okay? They also have a base on the bottom. This is the base. We need to carry them by the arm and by the base, okay? We will be using the stage to place our specimens, and we want to be sure that we don't turn the coarse adjustment knob too quickly, because we can easily crack a slide when we have the highest objective lens being utilized, okay? So really important, we want to take care of our equipment. That's the main thing. Okay, so now we're gonna look at a couple of slides here. We're gonna learn a little bit about the history of the microscope. Does anybody have questions before we get started? No? Okay, great. Here we go. Okay, history of the microscope. The first compound microscope was used around 1590, a long time ago, right? Think about the technological advances that we've had just in your lifetime, right? Did you all have, were there cell phones around when y'all were born? Yeah? But how big were they?
They were small. Yeah, they were flip
Really?
phones.
They were that small when y'all were born?
They were flip phones.
No, they were much bigger than that when y'all were born. But think about how far computers have come too. Cameras, okay? So the same concept applies here with the microscope. This was the first compound microscope. Right? Isn't that crazy? We've come a long way. Robert Hooke first used a compound microscope to observe pores in cork. So this is a cork slide. Cork is a plant material, okay? So he saw these and he identified all the little small boxes that he saw here with his compound microscope. And he decided that he was going to use the term cells to describe those, okay? These little squares. He called them cells. Leeuwenhoek, arguably one of the most influential scientist that has ever been around, okay? So many theories that we now use in everyday life, like the cell theory, the germ theory, were all influenced by Leeuwenhoek. So he was the first person to see single-celled organisms in pond water. He took those samples and was able to use a different compound microscope than what Hooke used. He actually created multiple different kinds of microscopes so that he could look at individual organisms. So there are some specimens that are best seen under a compound microscope, and then there are some specimens that are best seen under some different types of compound microscopes, which we'll talk about here in a minute. So you have a compound-like microscope. Like I mentioned earlier, it was the first of the microscopes to ever be invented. And then, can anybody guess or have an idea what another type of microscope might be? No? Nobody wants to guess? Okay. It is going I'm sorry, the electron microscope, okay? So electron microscopes are used to look at very, very small particles. We can see much more in depth with electron microscopes than we can with a regular compound microscope. You can see a lot of the details that you can't see with a compound microscope. So we're able to look at viruses, DNA, parts of cells. A lot of times I have students ask me, like, how do you know what that organelle looks like? How do you know what the shapes are? How do you know that's there? And we're able to do that by looking at images with an electron microscope. So the difference here is compound microscopes show you use in color, but an electron microscope is always going to be black and white. They have to go and add color in after the fact, okay? So it shoots beams of light and reflects those, it shoots beams of electrons rather than the light that a compound microscope does. So the light in a compound microscope is going to come from right here, okay? Much more powerful, much more detailed photo. A lot different than this, right? Yep, okay. Transmission electron microscope, much of the same concept as a scanning electron microscope. We can see magnifications up to 250,000 times. Wow. Imagine the end of this paper clip, right? Magnified 250,000 times. Okay, so we're going to actually look at the compound light microscope and identify some of the pieces and the parts to it, okay? So like I mentioned earlier, you always carry it with two hands. One arm on the base and one hand on the arm and one hand on the base, okay? Do not use like paper towels or anything like that to clean the lenses. If you ask me, I can get you a specific type of lens cleaning cloth. Don't force the knob. Like I said, if you turn this coarse adjustment knob too quickly, it will shatter the slide, okay? And we always store our microscopes covered in our cabinet so that we're sure to take care of them, okay? So, a little bit of information about the different parts of the microscope, okay? So this right here is going to be our eyepiece. It actually comes out and we have a lens cap cover that goes there when we're not using our microscope so that it protects the lens. This is going to be our body tube right here. This is your revolving nose piece, okay? And it's called the revolving nose piece because it turns. These are your objective lenses. So we have a 10x, a 40x, and a 100x objective lens here. As I mentioned earlier, this is the arm. These clips right here on the side, they're called stage clips, and they're used to hold the slide in place. So if I have a slide, and I'll spin my coarse objective knob down, and so I am able to secure my slide to my microscope. Okay. That way we ensure nothing happens to our slide. It doesn't fall off, it doesn't get broken or anything. So the stage clips are very important. This is the stage, okay? Stage, stage clips, stage, okay? Easy to remember, right? So down here you have your diaphragm. And so this is going to be controlling how much light you allow through from the light source down here, okay? You can control how much light based on the coloring and the clarity of your specimen. So that's really important. So this is going to be our coarse objective knob. This is also going to be our fine objective knob. This microscope is a little bit different than the one that we have pictured here. So I think that covers it. Like I said, this is our light source. Does anybody have questions before we move on about any parts of the microscope? A question? Okay, can you please tell me the best way to carry this microscope? Should I grab it up here by the eyepiece? No. Should I grab it by the stage?
No, ma'am.
Should I hold it with one hand?
No, ma'am.
No, I should be holding it with two hands, one on the base and one on the arm, correct? Great. And also, if you're moving the microscope on a table, don't slide it because it'll bump it and it'll mess up the stage, okay? So make sure you pick it up and move it instead of dragging it. That's very important. Okay. Okay, so magnification is going to be the increase of the object size. So whatever you have here on your slide as your specimen, using your objective lenses, that's going to change the magnification. Resolution is how detailed it is, how clear it is. Think about when you look at your phone, right? If you have a picture that you take that's really crisp and clear and you can see everything that's going on, that's high resolution. But if you are taking a picture with your phone and you shift really quickly, that picture's real blurry, right? Okay, that's low resolution. So we want high resolution when we're looking at specimens under the microscope. Okay, so both are needed to see a very clear image. That's what we're going for, okay, is a very clear image. So, as I mentioned, we have different objective lenses here, 4x, 10x, and 100x, and so that changes our magnification. So when we start using our microscopes, we want to make sure that all backpacks are out of the aisles. You can hook them onto the end of your desk right there so that no one trips when they're carrying specimens or carrying a microscope, okay? So you'll plug your microscope in. In some cases, we do have some that are just battery powered, so you don't have to worry about the outlets. We will store them with the cord wrapped around them and in the case and covered, okay? Like I mentioned, carry by the base and the arm with both